So what questions you have that haven't been answered, and just in a broad sense. So what do we need to know about Castleman's disease?
Like in my mom's system, right? So she, I guess she was classified as a normal before, but could something like another autoimmune disease called
Yes, that's a really good question. So that's asking about what's the natural and that's actually something that one of our studies is asking. What are the triggers for your flares? And so that's the question you're getting at. It's something that we look at. Anyone else?
So he's unicentric, but it's not resectable, so it can be taken out. So just dealing with the disease as a unicentric disease, but it's there, present, what to do other than wait and watch, so to speak. And then I guess also just kids with Castleman's over the long haul. And probably not known yet, but as we deal with that for a long case.
Yeah, and that's something that our registry is directly focusing on. So I'm just going to go ahead and go straight to this, which kind of gives an overview of the 5 buckets that I feel like all of our studies fall into. So the big bucket, the first one is looking retrospectively. So using information that's being generated from the clinics, combining it across patients, and then looking to see who's affected by Castleman, what symptoms are they experiencing, and how do they respond to different treatments. So what is the natural progression of the disease? How often does it occur in minors versus in different ages? So following buckets are all things that are more proactive. So one big area that I think would really help Castleman's disease would be to have a biomarker that you could directly measure and say, this person definitively has Castleman's, as opposed to excluding every other disease before we get that diagnosis. So this will dramatically speed up the time to get answers to the patients. We also want to improve treatment outcomes, so we're looking at who's currently receiving what treatments, how that correlates with their disease presentation, and how that results in different outcomes. Of course, we want to know what causes Castleman's, and this is where a lot of our efforts are focused right now. And the last bucket we're looking at is alternative therapies for Castleman. So, David has alluded to this, Mary alluded to this. We're starting to run a clinical trial now on a new compound, and so we're, very excited about this because this is really what's going to help patients. So, what's the best way to answer these questions?
Research.
There you go.
Everything we can give you from us.
That's wonderful. Yeah, so Castleman's disease doesn't have any model systems to study. So there's no Castleman's cell that you can look at in the lab. There's no Castleman's mouse. So really, it's the patients. They're critical to allowing us to ask these questions. So without hearing from you and understanding what questions you have, and then being able to basically look at your records, look at your samples, this is how we're going to get to the bottom of this disease. So to do this, the CDCN formed its network of patients, physicians, and researchers. We've been focusing on building infrastructure. So this is Accelerate the Patient Registry, which is extremely valuable, and then also our biobank, which we're launching now. So this is going to collect samples from Castleman's patients across the world. It's going to put them in one central location and make them available to any researchers who have important questions that they want to ask about Castleman's disease. So we're trying to make these resources available to the wider research community, so everyone can share them. And so this is going to allow us to understand the natural history of Castleman's, identify biomarkers, and then generate the next generation of therapies. So the first research program I want to talk to you about in more detail is Accelerate Registry. And so this is really getting at this first bucket of questions. Who's affected by Castleman's disease, how the disease progresses, what treatments work for it. So this is a study that anyone can enroll in as long as you have the word Castleman on your path report. You do it online. It takes about 20 minutes. So it's the first ever full patient registry. And what we're trying to do here is just combine records across 500 patients. this will allow us to identify subtle patterns, any sort of clues that may help physicians then better treat Castleman's disease. And this has actually allowed us to look at our diagnostic criteria and look at how patients are responding to therapies, and then provide that information out to physicians. Okay. Sorry, I got a question. So to participate, you can just go online, complete the consent. It takes about 20 to 30 minutes to answer questions, and then you have the option to complete a survey every 3 months. And this is the page you'll see. We have many members of our team. If you have any questions, please feel free to reach out. They can help you with this process. As David had mentioned earlier, we have about 150 participants, so we're making great progress. We're a third of the way there almost. We can do this. From these 150, we have about 80 complete medical records entered. And so each one of these records, there's about 2,000 different data points that we're pulling out and manually putting into a database. So this is a massive undertaking. When you register, that starts a whole process of weeks of work on our side. So we're taking this very seriously and really putting a lot of effort into it. So the results to date, we've captured about 30 different treatments and their outcomes. So this goes back to your question. And this allows us to start seeing associations between how patients are presenting and what treatments are working for those patients. So we're seeing the beginnings of a signal, and we're hoping that if we can enroll more participants, then we can go ahead and verify that the signal is there, and this will help inform treatment. Any questions on that? All right, and this next one is you probably could tell earlier, is the one that I'm most excited about. So how can we positively diagnose Castleman's disease? So Dr. Van Ree, who presented this slide earlier to show the overlap of all these diseases that you have to exclude before you can actually definitively diagnose Castleman's. So it shares features with autoimmune disorders, with infection, as well as with malignancies. And it's a cartoonish depiction of how IMCD kind of sits at the center of all three of these. So to ask if that depiction is accurate, what we're doing is we're actually looking at the blood of Castleman's patients in FLAIR and comparing that to blood from healthy individuals and people suffering from these related diseases in that Venn diagram. So as you can see here, sorry, got cut off. Here's a Castleman sample. These are what's present in the blood. Over here is the healthy or the related diseases. And you can see there's something in this blood sample that isn't in this one. And that's really what we're trying to identify. So to do this, we're looking at 1,300 different markers that are in the blood using a high-throughput platform. And we've assayed 92 IMCD samples for FLAIR, 44 healthy individuals, and 60 individuals with related diseases. So the next area I want to talk about is how to improve treatment outcomes. So Dr. Van Ree spoke about this paper earlier. And this is a very important undertaking for the field because for the first time, it's now memorialized how to treat a Castleman's patient, an IMCD patient. And so any physician anywhere in the world can find this online when a patient presents to them and get information on how they should proceed with treating them or know who to reach out to, whereas before it wasn't obvious. There was no resource. So as far as improving patient outcomes, this is going to have a very important effect. The other paper that Dr. Van Ree already presented, I just want to mention really quickly, is this predictors of response IL-6 therapy. So this is a very important publication as well that we published this year. And it's just getting to the point of we don't know who's going to respond to what therapies, but if we can find indicators that will give you a feeling of that the physician give them an answer if this patient will or won't respond, then they'll know I should try IL-6 or I should try some other approach. And so this will help inform physicians about how they should treat their patients once they're diagnosed. So the biggest bucket of our research program is definitely, the most of our research program is definitely focused on this question of what causes Castleman's disease. So what is the trigger? What could be the underlying pathogenic process that's responsible? So as David had mentioned earlier, Castleman's disease is really your immune system out of balance where it's become overactivated. So that's shown here. But why does this happen? So we have a variety of different hypotheses about why this might occur. One of them is that there's a virus. So this is really inspired by the observation that HHV8 can cause Castleman's disease in immunosuppressed individuals. So maybe there's an undiscovered virus that's causing it in IMC. Our second hypothesis is kind of it's analogous to a cancer model where you've acquired a mutation either through an environmental insult or for some other reason, and that mutation in a handful of cells is driving the disease. This is in contrast to an inherited mutation, which would be in all your cells throughout your body, and you would have inherited from one of your parents or both of them. And the last hypothesis is this autoimmune hypothesis. And this is getting at the idea that there could be a lot of antibody produced by some of the cells in your body, which are then attacking the other cells and causing your immune system to become activated. So for the viral hypothesis, this was we really favored this idea because it made so much sense compared to the HHV8 data that we've seen. So what we did is we decided to search for a novel virus in HHV8 negative IMCD and unicentric cases. We used technology. We actually collaborated with folks out of Columbia. It's the same technology that was used to discover West Nile virus. So this spans all known and unknown viral sequences. The samples we looked at were lymph nodes, so this is the area that you see HHV8 in, and HHV8 positive Castleman's disease. And the results that came back weren't what we were necessarily expecting. We actually didn't see any sort of novel virus in the tissues. So this basically indicates that it isn't a virus that's driving this disease. It's something that's innate or some other pathogen. So it could be bacterial, it could be fungal, it could be something else, but it's not viral. So we're currently in the process of preparing this manuscript for publication. The second two hypotheses will actually be, what was that? The second two hypotheses will actually be presented by two of our grant awardees following my talk here, so I'm not going to go into detail on them. But I will mention on the third hypothesis, the autoimmune hypothesis, that the CDCN has now raised $100,000. We're getting in the process of identifying the investigator that will receive this funding, and we're in the process of collecting samples from patients during flare to test this hypothesis. So we're making progress on all these. Obviously, research it takes a lot of time and effort and to generate the knowledge, but we are making progress. And lastly, I want to talk a little bit about the alternate therapies for those who aren't responding to asymptomatic. So we got really interested in a pathway called the PI3 kinase, AKT/mTOR pathway, for a variety of reasons. But basically, this pathway is an intracellular pathway. David had spoken about these earlier. So this is activity that's going on within the cell that's telling the cell what to do. And this is really important, specifically in T cells. It leads to their population expansion, so T cell proliferation. It leads to their activation. And a lot of these things that we're seeing in Castleman's patients can be explained by activation of this pathway. We're very interested in it because a drug exists that blocks it. So it's great to be interested in the pathway, but this one's especially interesting because we have this wonderful tool. So to determine if this pathway is actually activated in Castleman's patients, what we did is we stained lymph node tissue. So the brown here is an indicator of this pathway's activation. So this is a lymph node from a healthy individual. This is a lymph node from a Castleman's individual. And it's pretty black and white there, right? So this pathway is extremely activated in Castleman's disease. And we've done this now across 20 patients. It's pretty much every patient we've looked at is like this. So one patient who's received the sirolimus compound already had 5 different flare events. His remissions, the first 4 of them, the average was about a 10-month remission. This patient is now on a 60-month remission on this compound. So we have preliminary data from a handful of patients. It looks extremely promising. So as was mentioned earlier, David had applied to NIH and successfully received an NIH grant to fund the first ever federally funded clinical trial for Castleman's disease. And so we're going to be looking at this compound in more patients. So I didn't have enough time to go into all our different studies, but I did want to give you a flavor of some of the other things that we're doing. So these are a variety of studies where we're looking at patient samples and flare and remission. We're looking to see what cell types are present in the blood and what their activation status is, what they're doing. We're also looking to see how they're talking to one another and what else they're interacting with. So there's ways that you there's assays you can run to see what the immune system is seeing and what it's responding to. I mean, they're called T cell and B cell receptors. So these will physically bind to things in your body that have been either foreign pathogens that have invaded your body, or sometimes self-antigens, and this will cause immune cell activation. So we're very interested in those. We're also looking at gene expression. So this will tell us what intracellular pathways are activated, how active they are. And this is actually in a cell-by-cell manner. So we can tell you in this cell this pathway is activated. And the last thing that we're trying to do is allow pathologists to diagnose Castleman's more consistently and easier by analyzing a large number of bone marrow lymph node changes, and then actually training AI programs to identify what Castleman's pathology is. In this way, patients won't have to have multiple consults before somebody recognizes that they have Castleman's. That there will be this tool that every pathologist can leverage and use to see if the patient has Castleman's or not. So with that, I want to say thank you, and I'm happy to answer any questions you guys have.
One question.
In terms, knowing that it's not viral, what does that really help us know? Like, what are the facts that we know from it?
Yeah, so it rules out one very likely cause, and it means that we need to look at the other potential causes. So those could be, it's responding to some sort of self-reaction. So for some reason your immune system is recognizing something that's your own body that it shouldn't. And so that's very common in autoimmune diseases. And so we think that something like that could be driving this. Alternatively, there's a variety of different causes, and we just have to go through and kind of systematically test each one. But you have to start somewhere, right? And this was the one that seemed most obvious, so we started there.
Because like when I hear that, my thought is from like, okay, so when something is a virus, it can go from one person to another. Obviously, the afternoon, we know now that means having a virus would ever transfer from one person to another.
So it could be, so an HHV8 positive Castleman's disease, it's viral infection in the context of immunosuppression. So it could be that there was another virus in the context of immunosuppression or some other factor, but it would have to be kind of like a two-hit hypothesis.
And just to follow up on that, there's never been any cases of transmission of Castleman's disease historically. Cool. Well, thank you so much, Dustin. That was awesome. Glad he was able to go through the